Hello, good afternoon, everyone, and welcome to today's webinar. My name is Eleni Tokali, Drugs Intelligence Analyst at Global Data, and I will be your host. It is a pleasure to have you with us today to discuss Unlocking ADC Success, a hands-on tour of Turbine's virtual lab. I would like to welcome you to our webinar platform. This webinar is browser-based, so if you disconnect, click the link you receive via email to rejoin the session. We would encourage you to ask any questions using the Q&A widget or by using the Ask a Question box. We will try to get through as many of these as possible once the demonstration of the live lab has concluded. Please use the help widget if you experience any technical difficulties or require any more assistance, and please use the related content box to access additional resources. This webinar is brought to you by Turbine, a company that has spent the past decade building the world's first virtual lab for simulating disease biology. Turbine's virtual lab lets scientists run experiments on computers using a vast virtual library of cell lines, PDX, and even patient samples. This means researchers can test ideas and make discoveries much faster and more efficiently before setting foot in the wet lab. Turbine technology has already been proven through partnerships with leading pharmaceutical and biotech companies, including Bayer, AstraZeneca, Ono, and Cancer Research Horizons. Today marks an exciting turning point for Turbine. What started as a technology used only in select partnerships is now becoming an open platform. With the new virtual lab, scientists everywhere can design and run experiments quickly and accurately across diseases and cell types, and potentially even beyond oncology. Thank you for being here as we explore this new era of scientific discovery together. Today's guest speaker is Akos Tarksey, product manager at Turbine, where he leads the development of innovative digital tools for cancer drug discovery. He holds a PhD in chemical engineering from Budapest University of Technology and Economics and brings deep experience from roles at Chemaxon and Gideon Richter. Aka specializes in translating scientific and customer needs into actionable product features for AI-driven platforms. I would like to start the webinar by giving a quick overview of some of the ADC development challenges and what we at Global Data have been analyzing over the past few years. Let's start by looking at some of the key challenges in ADC development that have led to the current gaps in clinical success. One of the most critical challenges in selecting the right payloads or combination for ADCs. Payloads interact with cells in complex ways, and finding the ideal combination is not straightforward. So it's crucial to strike the right balance to ensure maximum therapeutic effect while minimizing unwanted side effects. Secondly, another major challenge is toxicity. Proper engineering of ADCs is really important to avoid causing significant toxicity in healthy tissues, which then limits its therapeutic potential. Finally, I'd like to touch on the biomarker complexity. Identifying and validating predictive biomarkers for ADCs remains a major hurdle, particularly given tumor heterogeneity and dynamic antigen expression. This challenge continues to impact patient selection strategies as well as trial design. So in an ideal ADC development process, understanding how payloads and their combinations will affect disease cells is the first step. Once we have identified the ideal payloads or combinations for a specific patient cohort, the next step is designing the molecule itself. This means selecting the right antibody or multiple antibodies and optimizing the linker to ensure that ADCs are targeted precisely and the payloads are released as intended, limiting toxicity. Now, let's zoom in on a small but really intriguing subgroup within the pipeline, the dual payload ADCs. Out of about 1,500 active ADCs in development, there are currently 33 that employ this combination payload approach. It's a tiny fraction, but it's a space that's growing and drawing a lot of curiosity. By delivering two mechanisms of actions in a single molecule, we get the chance to attack the tumor from different angles and reduce the chance of developing resistance. Designing these drugs, however, is not a simple task. It's a delicate puzzle, and we have to choose the right payload combination, match it to a target, and figure out a linker that lets both components do their job effectively. The potential payoff is huge if we can solve these challenges. This is exactly where simulation-based platforms like Turbines can make a strategic difference, enabling more rational payload selection, better design iteration, and potentially a faster path from concept to clinic. I would now like to welcome Akos, who will demonstrate Turbine's Payload Selector, an advanced tool built on a foundation model-like architecture and powered by virtual cells. He will walk us through the user experience and show us how the platform enables rapid simulation-driven selection and optimization of ADC payloads and their combinations across thousands of virtual samples of different preclinical model systems. Akos, over to you.
Thank you, Eleni, for the introduction, and thank you for joining me today to explore Turbine's virtual lab and especially the ADC payload product within it. The company's driver is to have positive impact on patients' life by supporting drug discovery with interpretable cell simulation. Turbine is successfully chasing this objective for more than 9 years now, run by the original founders and completing projects with more than 10 collaborators, including AstraZeneca, ONU, Bayer, Cancer Research Horizons, DeBioPharm, among various others. Now, with more than 75 people, we are working together to achieve this goal. But let's focus on one of the assets Turbine is building to achieve its goal, which is the ADC Payload Selector to generate biological insights to increase the success of experiments during payload and indication pair optimization. We have selected the cytotoxic drug, the warhead, that is responsible for destroying the cancer cells as our first scope within our applications. Since we realized that payload ranking, payload selection, and dual payload combinations are still challenging, and based on discussions with our clients and the scientific community, we realized that the dual payload approach is a fruitful new avenue that the different pharma companies are looking at to be able to deliver successful clinical results. Therefore, we tested our application on the large-scale pan-cancer cell line screening dataset that was published last year, measured on more than 700 cell lines and 100 drug combinations. From this dataset, we used the synergistic combinations as well as the single drug, the monodrug results, but excluded SN38 and its combination partners to be able to test out how our method is capable of predicting the synergistic effects. What you can see on the right-hand side is the simulation results compared to in vitro measured data for SN38 combined with olaparib. The x-axis is the HSA-access score measured in vitro, while the y-axis is the predicted in silico result. Each dot is an indication where the HSA access score was aggregated using max aggregation. What you see on this chart is that indications ranked at top by the model also proved to have high synergy in vitro, while those cases that showed less level of synergy were predicted to be also lower in terms of this synergy. So these encouraging results prompted us to develop our method further, and we tested various aspects of the model and presented our results at World ADC conferences last time in Asia. You can download the poster and check out our detailed scientific results. That also includes the sensitivity, prediction, accuracy on 5 payloads and on MMAE, DXD, and DM1 that were measured in Turbine's lab, and we used the test dataset to show our correlation values. The poster also discusses how this can be used to rank indications for a given payload. The second part of the poster deals with the synergy prediction, including how we prepared and cleaned the dataset and some additional results on this indication ranking for dual payloads. And the last part highlights the capability of the Turbine platform in terms of biomarker search, where we KO'd a large number of genes when measured and simulated SN38 sensitivity landscape, and identified schlafen-11 as one of the genes that is causing resistance in our simulations when comparing SN38 alone on large number of cell lines. And when these cell lines were KO'd for schlafen-11, we observed a drop in the IC50 values, meaning that when the schlafen-11 is KO'd, the cells became resistant. And this resistance mechanism was also traced back to one of the cornerstones of our method, that is the protein-protein interaction network. And we explored how the simulation took into account this protein-protein interaction network, and we could trace it back to signal propagation in the network, and figured out a mechanism that was also published in the scientific literature. So if you're interested, contact us or download our poster. With that, our model was shown to understand payload response and resistance to maximize the probability of clinical success in various use cases, including payload biomarker identification for patient selection, matching payloads with responsive cancer subtypes, design rational payload combinations, as well as understanding mechanistic details. We are actually moving forward with this ADC development track in a way that we are utilizing Turbine's wet lab capability to generate further data points to strengthen our model. As I mentioned earlier, we started measurements on payloads, and that led to extension of the payload library with additional payloads, and we are also investigating additional mechanism of action and additional drugs like alpha-amanitin, and now we are working with degraders as well to generate data and to be able to extend the applicability domain of our application. June, July, we plan to screen payload combinations and generate a data package that will be fed into the model to enhance the synergy prediction capability, while the lab will focus in August and September in antibody drug conjugate screening using ADCs from various sources, commercial. We are synthesizing our own small library of ADCs that is consistent and that can best generate data points that can be best utilized in model training phase. And we also acquire data from other CROs to be able to generate a seed package that is sufficient to start building antibody drug conjugate model that takes into account both the payload, the linker, the DAR, the antibody, as well as the antibody antigen interactions. And when the lab finishes the ADC screening, it will focus on the biosample space extension, including PDO and PDX data generation. While we are working on one of the tracks in the laboratory to generate the data, and in parallel the scientific team works on the model, we are extending the virtual lab capabilities by releasing some of the capabilities that are already available in Turbine platform to the virtual lab environment for external users to be used, including drug modifier CRISPR screen, where next to a drug dose response curve, a gene can be KO'd, and then the two sensitivity curves or sensitivity arrays can be compared to each other. But we also plan to improve on statistics and provide advanced statistical tools. The first wave of interpretation toolkits that we use in-house will be rolled out during late summer, as well as we plan to add a reliability score. So the application now can be used in two flavors. One is the subscription, where companies can subscribe with a given number of users who can run simulations on more than 1,000 cell lines, with perturbations on more than 100 drugs, including 15 payloads. And this payload count will increase in the future with our new proprietary dataset, what we generated. And then cell lines can be subsetted or filtered down to those cells that are having high expression for a given antigen, to be able to see for a special subset that is mostly interesting for one of the companies, and then rank payloads or payload combinations. Next to the subscription, which is a software as a service out-of-the-box solution, we also offer collaborations where we can extend most of these capabilities. And one thing what I want to highlight is that within a collaboration, we can work with partners and configure additional payloads and extend the list of modelable payloads so that they can be included in a payload ranking or in a synergy prediction, to be able to figure out which of the two drugs would have the highest effect in different indications. We also developed a more detailed ADC positioning package that is available within collaboration, that relies on TCGA and the protein cell surface expression data. So it's a more depth analysis under the hood. And we can leverage our internal capabilities like node and pathway level mechanistic interpretation tools in a scientific project. But if a payload is not characterized well, our wet lab capability can offer measurements to generate additional data points that we can use in the model training phase. And our wet lab capability can also be utilized to validate the findings of the screening results that we generated. So in case of a collaboration, we can move from ADC 1.0 version to a partner customized model that includes more payloads, and where we can broaden the scope of the application. But before going into a subscription or a collaboration, we offer a free demo access to our system to be able to have a hands-on experience by you, where you will also be able to simulate more than 1,000 cell lines, but in this case these are anonymized, on 16 drugs, including 7 payloads on the demo version, where you can try out how the results are generated, how the built-in analysis looks, and how it delivers insights and scientific results that can be turned into a scientific hypothesis. So how can you access the virtual lab after this webinar? I ask you to register at platform.turbine.ai/registration, and you can find the link via this QR code. After registration, your account will be activated within a week, and then you will be able to log in at platform.turbine.ai. Setting up a simulation is easy, fast, and straightforward. There are 2 major steps. You have to select the biosample space after selecting the model, define which of the cell lines you are interested in, and in the next step you should define the perturbation, in our case a drug or list of drugs, if you want to compare them, how their sensitivity trends look like across different indications, or if you select a set of drugs as main drugs and another set of drugs as combination drugs, then under the hood we will generate the cross product of this matrix and calculate on the given doses all the combination individual data points. And these data points are pre-processed in refinements that are run under the hood automatically to get to the more exciting part of the application, which is the analysis, where you can rank drug pairs, where you can align drugs with various indications or select synergistic combinations. So with that, I now want to move to the application itself, and welcome in Turbine's virtual lab. If you enter the virtual lab, you can see all your previous runs listed, and you have a browsing capability, so you can identify your previous runs even with a given drug run by you. Since I mentioned, the subscription is per company, so all users are assigned to a company now. I'm a member of Turbine.ai, and those users who are working in the same company can see each other's simulations and can analyze their results as well. However, companies are firewalled from each other, so no data can be accessed that's generated out of your company. So in the left-hand side, you see the filters, and this is like an ELN where you can see previous experiments. And if you click on one of the experiments, you can go into the details page, get an overview of how the simulation was set up, and then start the exploration. Now I will go back to the history view, and from the history, I will click the Create button to be able to launch a new simulation together today. So the Turbine Virtual Lab is a platform or a vehicle where we want to deliver you different models from Turbine and different capabilities. One of them, which is the focus for today, is the ADC Payload Selector, where we have the demo version available and the version 1.0. Demo version includes less payloads, as I mentioned, and the cell line names are not revealed, while in version 1.0, which is available with subscription, you can predict by also seeing the cell lines. We're also working on a clinical positioning bespoke model that will come with different options, feature sets, and that's for a different workflow. And also the company is active in target identification, where we use our internal platform capabilities, but we plan to roll out that feature set as well later on. So the first step is to select your model. Then, when your model is selected, you can go to biosample selection. As you can see, we have a wide range of cell lines available. As I mentioned, more than 1,000 cell lines are available. These are the in silico avatars. These cell lines are configured in our system, and they will behave as their fold change values, as their genetics dictate, since we trained these individual avatars in our model. The main table contains the name of these cell lines, lineage, subtype, growth properties, mutations, to help you figure out which ones you are interested in the most. So all the metadata is coming from the in vitro cell lines. You can easily filter down to some of the lineages, like I can add now some of the solid tumors, but we can also have myeloid, for example, or lymphoid. So within these 5 indications, as you can see, now we have 467 cell lines. So applying these filters will narrow down the list, and then you can pick one by one if you want to decide, or if you want to have a smaller set, but you can add all. If you want to run a pan-cancer screening without limiting the indications or lineages, you can go ahead and just select all the cell lines available. If you have a list like cell lines that you can measure in the laboratory, you can utilize the add batch or copy names function. With that, you can specifically simulate those cells that you have in-house. Next up, we have to define the drugs that we want to simulate, where we have this more than 100 drugs right now in the system. You can easily narrow it down to payloads. So you can add some of the payloads like MMEE, DXD, SN38. And since now I will set up a combination screening, you can add further drugs like FAT EGFR inhibitor, or I can look for checkpoint inhibitor and add those ones as well. So depending on your scientific objective, you can select the corresponding drugs to be simulated. So now I have selected 7 drugs so far, and I want to run a combination. So I will add further drugs to be combined, and as I mentioned, all by all combination will be evaluated. So now I will add DM1, DM4, MMEE, DXD, and we can see how this older payloads like SN38 vinblastine behaves as a comparison. But you can add further drug pairs as well. So if you want to have pairs that are not the well-known payloads, but completely new ones as well. So with that, you can easily set up your simulation. So now it is a 7x7 combination that we will screen, but I can add further elements from the list. When you are ready with that, you can go to the overview, and now as you see, we have these biosamples and all the details are listed here, or you can directly go to DepMap portal to learn more about the cell lines, and we have selected drugs for our simulation, where you can also check out the PubChem for more details, and these are the combi drugs. Drugs will be applied in the predefined doses. So these are the doses that we will apply. What you can change if you want to go with more potent molecules. So you can easily decrease or increase these doses. These are given in nanomolar. So it can be separately adjusted for the main drug and the combi drug as well. So now with that, we have 1 million, almost 1.9 million individual data points that we will simulate. And the last step is to hit the start button, where you prompted to a quick review, where you can check the biosamples again, lineages, drugs, combi drugs, and the total number of units that will be simulated. Here you can define your own name for the experiment, or you can enjoy the automatically generated name. So now I will hit the start button, and it will launch the simulation for us. A simulation at that scale takes about 30 to 60 minutes. And as you can see now, it is running in the background, so I can notify this message. And while this is being pre-processed, queued, and simulated, we will move back to the history view to analyze a previous run. And for that, I will select one from yesterday when I used the demo model. So this was a similar synergy prediction exercise. And that's the difference. As you can see, in this case, the cell line names are hidden. And you can also see that in this simulation we had these 5 drugs combined with these additional drugs in the combi simulation. When the simulation finished successfully, we activate the Explore by pages, where you will find the predefined dashboards with visualizations to be able to analyze your results. So after the simulation, in case of synergy, we are calculating the individual inhibitions and then combining these to calculate synergy scores. And the synergy scores, since now we have, as you see, the list of these drugs combined with the list of combination partners. And these are simulated across 467 cell lines. And on this heat map, what we are showing is the count of synergistic biological samples. So the purpose of this overview is to select from the possible large number of combinations those ones that are producing or yielding a higher level of synergy. The more red color is the lower number, while the more green color is the higher number. So what we can read from this chart is that vinblastine combined with romidepsin or SN38 resulted in quite high, or paclitaxel, quite high synergy, as well as SN38 combined with, for example, the V1 inhibitor resulted in high level of synergy. You can adjust by running filters the cell lines that are taken account when the heat map is calculated, and you can be more strict with the synergy window. So now I filtered out to 0.6, so only those cell lines are included now that are showing high level of synergy. Since if you go to FAQ, you will see that by default we consider synergy if the maximal HSA access score is greater than 0.3. So now if I go back, we can, with a more strict criteria, we can see a different overview of this heat map where SN38 vinblastine seems to have the highest level of synergy, and then maybe paclitaxel SN38, as you can see in this table. So our first objective was to pick from the possible combinations the one that is having the highest chance to result in synergistic combinations. So now I will go to synergy across drug combinations, and as we discussed, SN38 combined with vinblastine seems to have the best synergistic effect. And when I selected the main drug to be the SN38, we can compare again with its combination partners. And now I will release the synergy cutoff to the original value. So as you can see, both vinblastine and paclitaxel, so the top one microtubule inhibitor, seems to have a high level of average HSA score. And we also provide a breakdown based on the lineage metadata information. And as we see, vinblastine shows higher level of synergy in case of solid indications, lung, bowel, and breast, while it's moderate or lower in case of myeloids and lymphoid. So from that, we can move on to synergy across biological samples, where we are zooming into this given combination. So we started from a grid search across hundreds of cell lines, then we zoomed in to one of the pairs, and there you can continue with a detailed analysis, like how this is broken down into lineages as averages, or you can even go down to subtype level. So let's investigate together how in lung indication the different subtypes are distributed. So for example, here what I see is that on average or in median, the small cell lung cancer shows lower level of synergy compared to lung adenocarcinoma cell lines, and this is having some really extreme high values as well. So with that, you can also build up biological hypotheses, or you can rank even these subtypes across each other. And the biosample view, where you can also filter based on this HSA window to pick your best lineages. But it also shows you some parts of the raw data. So these are the doses from the main drug and the combi drug. And you can see the level of inhibition as the two concentrations are increasing, as well as the synergy, calculated synergy score changes. From that, you can figure out at which dose combination can you achieve the highest level of synergy. And you can even go beyond this and check for, let's say, individual biological sample. So now I will look at one of the bubble cell lines and see how the predicted inhibition grid on this matrix looks like, and what is the best combination, where we see subtle differences when this is administered at 10 to the power of -7, so 100 nanomolar, and the combination drug vinblastine is 10 nanomolar. So at those combination, we achieved the highest level of synergy. That might be useful for a DAR selection exercise. On the further tabs, you can even go into more details, and the general logic is that you apply filters. You start from a broad set and narrow it down, slice and dice the data sets until you find what you are looking for. The gene expression helps you to subsegment this into further categories. Now I will add ERBB2 as one of our genes, and we can investigate how SN13 vinblastine at different levels of ERBB2, HER2 expression levels distribute in terms of the synergy score. So for example, in lung, as we see, the high expression seems to have higher level of synergy. Yeah, in lymphoids, it seems that the low level resulted in higher level of synergy. So with that, you can combine your antigen interest with the results we generated so far. And we have also a means to investigate mutation effects, where you can go into the details, like if you want to compare wild type versus damaging mutations, and then on the box plots you can compare these categories, how in lung ERBB2 wild type and mutant cells were simulated in terms of the synergy. I don't have time to go into the details, but when the synergy calculation is done, similar to the drugs, the single drug results are also generated, where you can compare how the sensitivity landscape for the individual drugs that you included looks like, and compare their properties. But with that, I want to thank you for your attention, and I'm looking at the clock, so I will go back to the presentation for a second. And ask you again to follow the steps needed after this webinar. So to access the virtual lab, we invite you to register. You can use this QR code or platform.turbine.ai/registration and set up your first experiments. If you want to give us feedback, you can do it with this chat window, or reach out to us via email. We are really eager to hear your feedback and want to drive our developments based on your opinion and needs. And with that, I want to thank you. And now it's time for a couple of questions. So in the meantime, questions were posted, and I will read one or two of them since time is running. One of the questions is it possible to model proprietary payloads? So in the current system, in the subscription version, Turbine offers the number of or the set of payloads, but in a collaboration, we can use partners' data to configure the model and extend the payload set. And we need 10 to 20 measured viability IC50s for that at the very minimum, or we can help you with generating this data set. So for completely new payloads or proprietary payloads, we need a retraining, and that will end up in a scientific collaboration. There is a question: can I simulate payload response for high antigen expression cell lines? So in our model, we have the cell lines and their metadata is available. As I mentioned, you can filter down from a simulation to those ones that are having high expression, and by setting only those cells in the simulation, you can generate the data only for that part as well. And there is a last question. Do you only have cell lines available for testing? Use organoid models too. So yeah, we are aware of this, that now the industry is moving towards PDX, PDO, and we also want to follow this trend. Currently, to my knowledge, in our lab, we only support cell lines. So if you want to run a validation with us, then these are cell lines, but we have partners with whom we can also organize a PDX or PDO screening or organoid models as well. And as I mentioned on the roadmap, down the line later this year, we also plan to generate or acquire data on organoids and make the ADC capability and the payload capability available on those more sophisticated models as well. And as I see there are a bit of additional questions, we will come back to you in an email for the sake of time. And now I really want to thank for your attention. And I hope to see you in our software as a service or among our collaborators in the near future. And thank you again for your attention.
Thank you, Akos, for a really interesting walkthrough of the lab. Thank you again for your questions and to Akos for his answers. That just leaves me to thank you for a great demonstration and discussion, and to thank Turbine for sponsoring this session. To the attendees, you'll receive an email tomorrow telling you how you can access the on-demand version of this webinar, or you can access this through the website www.arena-international.com/events. Please continue to ask questions and interact with the widgets as the on-demand versions remain interactive. This just leaves me to thank you once again, and I hope you all have a wonderful day.